participate. I wish you good success for the conference. Now, it's my particular pleasure to introduce the presentation by Professor Skrynnikov. Excuse me, Professor Skrynnikov, that I do not mention all the co-workers of your presentation. I believe you will say about these details later. It's my particular pleasure to introduce your lecture because you introduced a new direction of research at the St. Petersburg University, and we know about the long tradition. We had a nice presentation by Professor Czichik, and now we are very interesting to look to the details of your lecture. For me, it's in particular interesting because you develop new methods, and on the other side, these methods are very important also for practical problems in medical sciences. So we are very interesting to listen to your and your co-workers' presentation. May I ask you to start now?
Yeah, thank you very much, Professor Michal. Thank you, Vladimir Ivanovich, for inviting me here. So let me grab the pointer. It's right here. I'm going to speak about some latest results from our laboratory, which is Laboratory of Biomolecular NMR at St. Petersburg State University. So in a way of a bit of a history introduction, the laboratory was set up late in 2013. There was a mega grant from St. Petersburg State University that we won. And for the last couple of years, we've been supported by the Russia Science Foundation. So I should give a credit to Vladimir Ivanovich, who is here. He was really the inspiration behind this laboratory. He got us together and persuaded us to write this grant application. And that's how all of this started. So we pursue a number of projects that are listed here. The header is the project by Sebastian Rabdana, who is here in the audience, and he's going to speak about his work. That's the next talk. And then Kerstin Kampf, who was a postdoc, came from Germany to stay with us for a year and a half. And she worked on a synthesis of experimental NMR, relaxation, NMR measurements, and molecular dynamics to study the motion of intrinsically disordered proteins. And then there is this paper by, or rather the project of Sergei Izmailov. So he worked on molecular dynamics modeling of the process of disulfide bond formation in peptides and proteins. And that paper is under revision in scientific reports. So we're just putting some finishing touches on the manuscript. Then there is a work on the rocking motion of the proteins in protein crystals, and that's something that I actually talked about at the last Spinos conference in late November. And this work was just accepted by Nature Communications, so we're very pleased with that. Sergei Izmailov and Olga Rogacheva are co-first authors on this paper. And Dmitry Luzik is the point person on the structural study of methyltransferase and SUN7. That's a collaboration with Olga Anatolievna Donsova in Moscow State. So that's a project that we are also happy about. And there is something else that also is spearheaded by Olga and Sergei. We think that we've discovered a new type of interaction in proteins. It's a sort of charged dipolar interaction between the charged lysine side chain group NH3+ and the carbonyl group. So for that we are writing a communication to Angevante. But the two things that I'm going to cover in this talk are the projects. One of them has to do with cell biology, so these are cell culture experiments. And we've applied NMR, but in an auxiliary role, I should say. And the other one is more of a hardcore NMR of amyloid fibrils. So I'm going to proceed with that. This is the story that started in, I believe it was late 2012. And I was walking across campus at Purdue University and bumped into my colleague, Professor Lau, who was heading to the talk given by this fellow. His name is Donald Keefe, and he works at Harvard Medical School. So he's one of those types who have both PhDs and MDs, and he is a practicing doctor. I attended the talk, and basically this was the starting point, and there was a period of time when I actually sort of regretted that I got into this business, but I think now by the end of the day I'm happy about it. It really opened some new horizons for myself personally and the laboratory. So a kind of the main direction of the Kiev's laboratory efforts over the last 15 years or so is the study of the protein which is called, it's epithelial glycoprotein mucin 1. And that's the schematic of this protein. So epithelial means epithelial tissues like the inside of the cheek, for instance. That's where it's mainly expressed. And if we look at the schematic, here is, if we can go over the entire length of the protein. This is the C terminus. It's inside the cell. It's a cytoplasmic domain. You can see it's disordered. It doesn't have any definitive structure, 72 amino acids. Then there is a stretch which goes across the membrane, has about 28 amino acids. There is 58 amino acids here, a little globular domain, which is kind of funny because it's not a continuous polypeptide chain. There is a cleavage point in there, but nevertheless it forms a unique fold, and by virtue of the hydrogen bonds and those like usual folding factors, it just kept together and serves as a sort of a lock. Which connects the transmembrane and cytoplasmic part to this ectoplasmic portion of the protein, which is really long, close to 1,000 residues. And it's heavily glycosylated, which means decorated by sugars. And the idea is that you have this layer of sugars on the, let's say, inside of the cheek. And what this provides is a protection against bacteria. So it's kind of mechanical protection that the bacteria can't really sail in and do whatever damage to the tissue. So that's the protein. Donald Kiev claimed that the cytoplasmic domain is really an important signaling portion of this protein. There is a whole lot of interactions. This is the androgen receptor. That's the biggest protein in prostate cancer that conceivably interacts with cytoplasmic domain of mucin 1. And FKB is a very famous oncogenic transcription factor. Likewise, DNA binding domain of STAT3. We've looked at it, we've expressed these proteins and failed to see any binding, so that was the disappointing part. In process of doing it, we've also obviously expressed this cytoplasmic domain of mucin 1 and made sure that when you look at this HSQC spectrum, you realize immediately that this portion is indeed disordered, because you can look at the amide protein chemical shifts and they all follow in this narrow range, and this means that kind of the spectrum collapses. And what this means really, it's random coil-like protein, and chemical shifts are not a whole lot different for one residue relative to the other residue. So this is how it is. And there was a continuation to this story. And that was really the focusing point of the efforts in the Kiev's lab. Here is another excerpt from the sequence of the mucin 1. And that's the portion that sticks outside the cell, and this is a transmembrane portion. So those are mostly hydrophobic residues. This is immediately recognizable. And this is the 72 amino acid cytoplasmic domain that extends inward inside the cell. And again, you can tell right away that these are hydrophilic and polar residues, and the prediction algorithms actually can tell you that this is a disordered sequence. Now, the belief is that the entire thing, mucin 1C, is functionally active when it's dimerized. And in order to dimerize, apparently the key role is played by this pair of cysteines. So presumably, putatively, there is two disulfide bridges formed here, and via those disulfide bridges, the protein can dimerize. So what Kiev has invented, his idea was he has designed a peptide which is continuously under development, so that's the latest generation of this peptide, which kind of mimics a portion of the mucin 1, and it's capable of forming a dimer, presumably. And by doing so, it blocks the dimerization site so that the real full-length dimer cannot form. That's the idea. And this is something that he tested in a therapeutic context, first in cell line experiments, then on animals. So these are the tumor xenografts in nude mice. And this axis here is the tumor volume, and these are the days, right? So the volume just increases almost exponentially. That's the uncontrolled growth of the tumor. But in those animals that are treated with the peptide, which are given the injections, you can see that actually the tumor shrinks and all but disappears. And this is, these are very impressive pictures, but by now I'm kind of used to seeing such pictures. This is the histological staining of the tissues. So that's the H and E staining, and this is a cancerous tissue, and this is after the treatment. You can recognize some tumor in here, but not a whole lot. So all of this seems, fine and dandy. Donald Keefe set up a company which is called Genus Oncology, raised $10 million to fund those studies. There are 3 clinical trials. One of them is already completed. The other is ongoing. They're recruiting patients. And the third one has been scheduled and now is canceled. And I don't know, but the chances that our publication might have something to do with this. Not likely, but can't be ruled out. So at one point, we've looked closer at this peptide. And there's one thing that we paid attention to. So as it happens, the peptide, the latest generation GO203 peptide, is synthesized not from L amino acids, which are natural amino acids, but from D amino acids, which are unnatural. So it's like life and death, L and D, right hand and left hand. And there is a good reason as to why, someone would like to synthesize this peptide from D amino acids, for the reason that this type of a peptide is less vulnerable to a proteolysis by proteolytic enzymes. So the L peptide is eventually, pretty soon, is going to be chewed up by all those enzymes, and this one is stable and is going to have a longer lifetime. But then thinking about it from the structural perspective, you realize that actually, this peptide, in terms of topology, has little to do with the original sequence. It's like left hands and right hands, right? If you try to put the glove from the right hand onto your left hand, that's not going to be comfortable. It's really, from a topological perspective, it's a different animal, different beast. And then we asked ourselves, okay, like this is the way it's spelled. These are the same letters, C, Q, and so forth, right? So from this perspective, you can argue that's going to produce this dimerization behavior. But if you look at the architecture, really, like those amino acids have nothing to do, almost nothing to do with those. And then what would be the basis for selectivity? Why is that this peptide is going to target specifically mucin? Really no basis. It could be actually, acting upon any target rather than mucin. And so this was this initial realization which made us start this project. We can take it one step further because, in a sense, Donald Keefe and what he was doing was limited. He thought he really needs a piece of the mucin sequence to get the peptide to work. He reduced it to a pretty short piece. It kind of became a little bit iffy if this short piece is sufficient for selective recognition. But he stuck with just copying the sequence. And we are no longer limited with this because so long as we realized that the peptide has little to do with mucin, we can start varying those amino acids one by one, hoping to enhance the anti-cancer activity. So that was our motivation in going into this project. And indeed, lo and behold, we've demonstrated that the effect from GO203 does not correlate with the expression of mucin 1. And I should mention here one thing. So mucin 1 is really a legitimate cancer target. It's very widely known. It's commonly recognized. And the reason why is because it's overexpressed on the surface of many cancer cells representing different cancer lines. And then once you know this fact is established, that opens the route for potential cancer therapy, because you can attack selectively mucin 1 and you can be assured that actually your drug is going to be delivered to cancer cells 1,000-fold more efficiently compared to normal cells. So it is a cancer target. It is a legitimate cancer target. It's just that this has little to do with the GOT03 peptide. So here is the confirmation. We had a number of cancer cells. This is fibrosarcoma. This is breast cancer. This is lung carcinoma. These are actually normal cells, dermal fibroblasts. These are the cells that grow into skin. And so here you recognize something which is called Western blot. And this Western blot tells us that the breast carcinoma, this specific cell line, actually does overexpress specifically mucin 1. Others not so much or not at all. And this kind of matches the literature evidence. This is right. But now if you look at how those cells respond to the treatment by the GO peptide, and you can look at the right portion of this bar diagram. So this is the dose of 5 micromoles per day, and this is 10 micromoles. And you see these are the cells overexpressing mucin, and they do respond, but kind of reluctantly. So even at a very high dose, many cancer cells survive. Unlike this fibrosarcoma thing, where already 5 micromoles actually take the toll on the cancer cells, and 10 micromoles virtually wipes it off, and then even higher dose, they are all dead, nothing is left. Then of course the side product of this experiment is our dermal fibroblasts. And you can see that they are also really badly damaged with this peptide. So really, it's not an anti-cancer peptide. It is a cytotoxic peptide. And those hapless patients that get up early in the morning and go to this hospital in New York to get the injection and so forth, they are being fooled in a sense, because really this is not an anti-cancer therapy, as it turns out. But we've continued investigating the mechanism of those peptides and learned a good deal of useful and interesting things. These are so-called dot plots obtained by cytofluorimetry. And the idea is you basically stain your cells with 2 different markers. This marker has to do with its detection of caspases 3-7. And caspases are very specialized proteins by means of which the cell basically shuts down, it self-destructs. So if the cell senses that things are going very wrong, and it decides to kind of surrender and just in an orderly fashion die. And there is this whole execution cascade that the cell self-destructs, and those enzymes just destroy all the protein machinery in the cell. And this is the other marker. This is the marker that binds to the double-stranded DNA. So this marker becomes activated when the nucleus ruptures and all the DNA comes out, and then you get a fluorescence out of it. So that's basically the dot plot which illustrates the process of cell death. And what happens with control cells, they are all healthy. They are all sitting, this is a low level of caspases, low levels of cytox. This is a healthy population. And when you expose it to the treatment by GO203, this is the advanced apoptosis. So they all traveled kind of through this path. First, the caspases kicked in and kind of chopped the proteins inside. And here the nucleus is ruptured and all the cells now are in this final death throes, which I'm going to also show this little movie from the confocal microscope. So that is called an apoptosis. And you can see like this is a healthy cell and it's nicely spread out. And then I suppose that's going to continue playing with some luck. And so what happens with this one is called blebbing. So to explain the meaning of this phenomenon, I'll tell you that there is a membrane of the cell, and underneath the membrane there is something which is called the cytoskeleton. So it's a meshwork of proteins and primarily actin and spectrin, and it gives the cells its elasticity because the cell needs to be elastic. And once those caspases are expressed in the cell, the caspases starts cutting through this meshwork, the actin meshwork, and the disruption of this meshwork causes the production of those blebs, like it starts really boiling on the surface, bubbling, and they kind of flow away. And I don't know if I can make it. I'll play further. Should be able to. But eventually what you're going to see is that this, look at this, like this one, instead of being nicely spread out, it kind of contracts and becomes rounded and more compact. So it's again the same thing, the effect of the caspase, and then the blebbing also starts here. So that is the effect of the GO203, and it actually kicks in pretty quickly. It's a pretty dramatic thing that you pipette this peptide, and in a matter of half of an hour, the cells start getting uncomfortable, and then eventually they die. So that's the second part of our agenda. We thought that we could redefine our peptides and design a new family of peptides and see how that's going to affect the response of the cells. And there are two approaches. One would be sort of combinatorial approach. Just take a peptide and start varying each amino acid one by one, then in combination, so forth, which would be pretty labor-intensive and pretty expensive. So we've chosen a different route. We've actually identified the sequences in the known proteins containing two cysteines side by side. So here you can barely see them probably, but it's a pair of proximal cysteines. And we do know that those cysteines are key to the cytotoxic activity of the peptide. So this was a sequence extracted from disulfide oxidase, from disulfide isomerase, from the reductase, and so forth. So we've designed a whole bunch. There were some metal binding motifs and so forth. And some of them did prove inactive, like for instance when you knock out those cysteines and replace them with similar serines. So serines are also small polar amino acids, but they're non-reactive. And you replace them with serines, and this is no longer active. So the cell population is more or less preserved here, even though you expose it to considerable dose of the peptide. But look at this, for instance, disulfide oxidase-like peptide right here, or disulfide isomerase peptide. They are just a good deal more active than this original peptide, which was devised by Keefe. And that actually tells us something about the mechanism. So that made us think that that's how the mechanism looks like. It's all in this little schematic. I'll start from the left side. And we need to recognize that there is a certain amount of oxidants in the cell. That's this famous oxidative stress that Sebastian is going to tell a little bit in his talk, I assume. So what's shown here is hydrogen peroxide, but also there are free radicals, there is superoxide and whatnot, right? And the primary target that's going to be actually addressed by those oxidants are the cysteines, in this case the cysteine residues belonging to the peptide, those two cysteines with their thiol groups that are originally in a reduced form, but under the effect of the oxidants they are going to cross-link with each other and form this disulfide bridge. This is what happens. Now this peptide has an ability to approach a protein and to kind of dock to the protein surface. Not very specifically, presumably. I mean, I don't see how the specificity is programmed in this peptide, and not very strongly, but it does have the ability to meaningfully interact with the protein. And when it interacts with the protein, which at this point we don't know the identity of the protein, let's say for a moment, something happens which is called the dithiol-disulfide exchange reaction. So you see this disulfide bridge opens up, and at the same time, two cysteine residues which were, unconnected, they were reduced, they become oxidized, and they form this disulfide. And this disulfide is not supposed to be there. It's non-native, and it disrupts, interferes with the function of the protein, and disrupts and compromises the structure. And basically, that's how the apoptosis and the cell death is triggered. So in a sense, the whole process is not a reflection of let me put it this way. Is it the role which is played by the peptide by itself is not a factor of oxidative stress. The peptide is a sort of mediator of oxidative stress or enhancer of oxidative stress, because what peroxide alone cannot do to the protein target, with the help from the peptide, it can actually achieve. It can produce this disulfide bridge, which damages the protein target. And so the paper came out recently in the scientific reports where we reported those findings. The reviewer asked us to provide more evidence towards this hypothetical mechanism. And Ivan Podhoretov has run those experiments, so I'll explain a little bit the meaning of these experiments. As an assay protein, we've used hen-egg lysozyme here. So Ivan reduced it, and hen-egg lysozyme contains actually 4 native disulfide bridges. So Ivan reduced it and denatured it and lyophilized it, and then you start with the denatured lysozyme as also random coil-like. So if you take the control sample and you run this control sample through the chromatography column, it comes out as it's this peak. It has been identified. This is a denatured lysozyme, and even after 15 minutes or 1 hour, still you get out this denatured lysozyme. But if you add a little bit of the GO202 peptide, then already after a minute, the denatured lysozyme is gone. You have all sorts of, species here where some disulfides are formed. And actually, after an hour, most of your lysozyme has folded in a native conformation. And so the way that we take it, this is a confirmation of this mechanism, because the peptide has the ability to mediate the formation of disulfide bridges. And what's shown here, it's kind of unidirectional. It only mediates oxidation. Presumably, it can also go the other way. So it can, depending on redox potential, it can also mediate the reduction of undesirable disulfides. So this is a test. You take it at a face value. This is really how the mechanism is. And I'll show you also some latest results that were obtained maybe a month ago or so. So this is yet another cell line. This is the liver carcinoma HepG2. And this is something interesting happens with the cells, with the morphology of the cells when you treat it with the peptide. So those are little colonies. They are sort of shaped like raspberries, and the cells are very compactly packed here. But when you treat it with the peptide, after 3 hours or so, the morphology becomes dramatically different. And for a while, we thought that this is something which is called epithelial to mesenchymal transition, which is really terrible, because this epithelial to mesenchymal transition means that the tumor cells, which are all localized and they're sitting tight, and they become capable of migrating. And this is a metastasis. This is metastasized cancer. That's what they thought. we started running those experiments to actually explore it in more depth, and this is the experiment which involves the stain for polymerized actin. So I've been bringing up this cytoskeleton, the meshwork. This is how it looks like in the normal cells. And this is how it looks like in the cells that have been treated by the peptide. So you can see the change is quite dramatic. There are those deposits of actin which are called stress fibers and so forth. And this is apparently responsible for the, for the outgrowth of the cells. So actin gets oxidized and gets abnormally polymerized. That's something that we are going to try to confirm now using in vitro experiments. So, as a take-home message from those experiments, you could ask, so you've discovered this cytotoxic peptide, and this is by no means unique. There are plenty of cytotoxic peptides. There are toxins and so forth. Can you actually cure cancer with that? And we have those experiments ongoing. So what you can do, you can actually prepend to this active cysteine-containing sequence the specific sequence that's going to serve as a homing and targeting device specifically for the cancer cells, so for receptors like GHRH and so forth that are also overexpressed on the surface of the cancer cells. So we'll see if we have any luck with that, maybe not. But anyways, these are small steps along this road. So now I'm going to switch the gear, and I'm going to talk about something that has more to do with NMR, which is really our base strength. This is the project that focuses on fibrils. And as you probably are well aware, fibrils is this unusual state of the protein where a protein first unfolds, and then typically it forms kind of hairpins, and those hairpins are then stacked on top of each other. It's like one hairpin, the other one, and so forth. And you get a fibril, and there is this uncontrolled growth. And all of this, unfortunately, happens in our brains, usually at advanced age. And eventually, the neurons are being overwhelmed by this growth of the fibrils, and they just die. And that's Alzheimer's disease, right? So that's a very sad event where the person's memory fails, and then person's intellect fails, and towards the end of the day, it's really like the person is reduced to the vegetable state and most likely died from pneumonia, having lost the ability to recognize his family and so forth. And while we actually make quite impressive inroads in our fight against cancer, the same cannot be said about Alzheimer. So if you ask me if there is any medication at all to treat Alzheimer's disease, the answer is zero. Like, you can take vitamins and you can take certain things to improve your blood circulation, which would make you feel better for a while, but then it's all down the hill and into darkness. And so then, obviously, there is this huge interest in studying fibrils. It's kind of almost a different phase state in the realm of proteins. And we had an opportunity to kind of jump on this bandwagon. So the fibril in question is the fibril which is formed by this SUP35P. That's from yeast, so that's дрожжи. Пекарские дрожжи, and it's actually a translation release factor originally, but it forms fibrils, and it also happens to be a prion protein. So if you heard about mad cow disease, Jakob-Creutzfeldt disease, that's an infectious protein. So the infection is not lethal for yeast, but as kind of like a flu in yeast terms. And in terms of sequence, so it's about 250 plus residues. Then there is one portion of it that's for certain forms fibrils. So this is this blue part of the protein, it actually kind of forms this type of assembly. And now the yellow portion of it that the question is up in the air. So Rob Tico of NIH, who really is a genius in this field, he proposed this model. So he thought that really the so-called M domain is an extension of the fibril. There are some positively charged amino acids, negatively charged amino acids that are stacked on top of each other, ta-da. But that remains controversial. Is it structured or is it flexible? So we set out to investigate it by means of NMR spectroscopy mainly. We've made those fibrils, and that's the collaboration with the people at the biology department, Mikhail Belousov, Stas Bondarev, and Galina Anatolyevna Zhuravleva. So Mikhail actually made the sample for us, and we make it form fibrils, and Ivan took this electron microscopy image, so we satisfied ourselves that these are fibrils. We were actually able to place this, this sample in the solution state NMR tube. We've also run some solid state NMR together with Sergei Vyacheslamovich Dvinskih that I could show separately, but the results that I'm going to talk about in the remaining part of the talk have been collected in the resource center under Pyotr Tolstoy 500 megahertz machine. It is not awfully difficult to actually detect the flexible tails, and that was the experiments that we ran. Our agenda was to try to detect the flexible tails of the fibrils. So let's say like here. One thing that actually not very many people realize, that any fibril is in fact a dynamic equilibrium. So there is a fibril per se, and there is a certain fraction of monomers floating around, and they are in a state of exchange. It's a very slow exchange with very high kinetic barrier, but it's a true dynamic equilibrium, and you just can't walk away from it. And so then there is an issue. You want to really discriminate between the tails, flexible tails of the fibril, and between your monomeric protein floating around, presumably also flexible and so forth, and you want to draw this distinction. And that's what we've done. And we've done it using the diffusion sorted experiments. So Ivan was really the person behind it, which are popular. I mean, they are popular at our department. Many people run those experiments. And let me just run those animations, which probably I needn't do in this audience. Like this is a simple cartoon illustrating spin echo effect. So there is chemical shift evolution, then the magnetization is flipped by means of the 180-degree pulse, and then it continues evolving under the chemical shift. And because two delays are carefully matched, right, by the end of the day, the magnetization is aligned here along minus y-axis, and so the magnetization is refocused, and that's spin echo, that's Hans echo. Now I'm going to introduce two elements here in this simplistic discussion. One is going to be the gradient. So normally your magnetic field obviously is manufactured such as to be highly, highly homogeneous to within a fraction of hertz, right, if you are shimmed properly. But here deliberately we kind of ruin this homogeneity. We create the inhomogeneous B0 field. So let's say at the top slice in the sample, the field could be 11.6 Tesla, and here 11.7, and here 11.8 in a bit of exaggerated fashion, but that's how it is. And so now the second thing that we need to bear in mind, that there is a diffusion that comes into play here. And that's schematically illustrated here. So again, chemical shift evolution, and again, then it's sort of the magnetization flies on the surface of a cone. But then recall that diffusion is present in the system. And it needs to be taken with a grain of salt, right? Because I kind of separated spin evolution from this Brownian diffusion. And in fact, they tend to, happen concurrently. But I think that conveys the idea. And so the molecule sort of moves randomly. And then there is I think, to be continued. It keeps moving. And then eventually we're going to watch the refocusing part, but now the refocusing is going to happen in the field, which is different. It's lower in this case than the original field. So coding gradient and a decoding gradient are separated by a period of time. And as I need to sort of drive it manually. But here is now this evolution. And you see that now this evolution is happening, well, you can appreciate that this, refocusing was happening in a lower field. It was happening at a slower pace. And then the vector kind of fell short. It failed to reach the minus y-axis. And then it doesn't take too much imagination. Here we've been just tracking one molecule, which was kind of drifting this way, right? But other molecules can go in different directions. And then the result is going to be that this refocusing process is going to be disrupted. So we'll end up with those magnetization vectors associated with the individual molecules kind of spread out in a circle, and then the average signal is going to be lower because of that. It's kind of dephasing that's associated with the diffusion in the inhomogeneous B0 field. And this is the experimental results from our system, our sample of SUP35NM. So this is a standard proton spectrum. As we increase the gradient strength, the losses become worse and worse. So you see the intensity of the spectrum kind of progressively decreases. But then after a certain point, it decreases no more. So you see those multiple colored traces are on top of each other. And here, if you actually plot the intensity as a function of gradient strength, here is how it looks like. And we have an explanation for that, because all those low molecular weight fractions like monomers and dimers, they really float around. Fibrils don't go anywhere. They are like elephants. They are massive. They are huge. They just stand still. And so the fibril magnetization is there. It's unaffected by gradients, unaffected by diffusion. So in this particular sample, we have about 15% of fibrils. And so that's the way to actually separate, to separate monomeric fraction from the fibril fraction and focus on the spectra of the fibrils. So obviously, Ivan hasn't stopped there, and he has undertaken this comprehensive analysis of all the data that he has collected with different diffusion delays from 30 milliseconds to 1 second. So that's still this gradient dependence, but the delay between the dephasing and rephasing gradients is varied. And you need to also take into consideration the different relaxation rates in the monomeric fraction and the fibril fraction. And in both cases, we are actually talking about those flexible tails. We do not see the body of the fibril that is reserved for solid-state NMR. But still, they're different. Monomer is really different from fibril because fibril is more dense, like, all those tails are kind of rubbing shoulder. It's a little bit locally more viscous environment. So relaxation rates are different. But once Ivan actually fitted the entirety of all this data to the same set of equations, which are like Skrynnikov equation for the diffusion experiment and so forth, he came up with those best fit values, and you see that they've been determined quite accurately. So this is from Monte Carlo. This is the diffusion coefficient for monomeric SUP35NM, exactly what we would expect for the protein of this size, about 200 times, 400 times slower than water. And then there are also the relaxation times for the monomeric and the fibril fraction, and the fraction of the monomer here is 50%, is a different sample, and everything fits all together and works. Now we can move a little bit one step further compared to the simple protein experiments. We can look at HSQC sequences. And we can insert those coding and decoding gradients in the HSQC. So briefly, the sequence works like this, that this is an inept element, right? And the magnetization is actually coded here by the gradients. And then we record the nitrogen-15 spin evolution here. We do it in a constant time fashion because we want to have this well-defined and constant diffusion delay. So that's a constant time kind of thing. And at the same time, the magnetization is actually converted into pure in-phase nitrogen. So that becomes NX magnetization. And then you choose to store it along the z-axis because then the relaxation losses are reduced. It's the longitudinal relaxation, as you all know, is much slower than transverse relaxation. And then you go backwards. You have a pair of inepts, and those are refocusing gradients. So then you sort of reconstruct your signal. So this is what we've done, and these are the spectra. So these are two-dimensional HSQCs, right? Again, what's recorded here is a protein chemical shift and nitrogen chemical shift. Now the blue spectrum is the spectrum with a very weak gradient strength, so it's essentially unfiltered, and it contains everything. It contains, monomers, or if there are some dimers and so forth, and the flexible tails of the fibrils. Everything goes in here. And what's shown in red color is a very strong gradient of 50 Gauss per centimeter. So then everything that's mobile and low molecular weight is filtered out, and what remains are the flexible tails of the fibril. And so this was surprising, and for a while we couldn't wrap our minds around it because the spectra are similar. And then we realized that here is what's going on. So if you look at the fibril, you actually see the C-terminal domain, the so-called M domain, the yellow domain. It's flexible. All the resonances are, belonging to this disordered domain. But if you actually look at the monomeric protein, it's a similar picture because apparently this behaves as a bona fide disordered protein with well-resolved resonances. But the N-terminal domain, it has this tendency to stick. It's really sticky. And it forms this kind of, inhomogeneous kind of globula here where you don't see spectral signals from it. So this is why, the sets of the spectral signals look similar. And we can actually, delineate the boundaries of this flexible part. So for this purpose, we made a sample which is labeled selectively with nitrogen-15 only in valent position. And to make this protein, you just need to take your bacteria and you feed it with a special media which only contains nitrogen-15 labeled valine. And then this valine is incorporated in the process of biosynthesis in a peptide chain, and you end up in this selectively valine labeled sample. And all those valence happen to be in the M domain. They all happen to be within this part. And lo and behold, we can see them in both spectra. So in unfiltered and in the filtered spectrum, you can see very well the red color here, but trust me, it's here everywhere. So all those resonances belong to the flexible portion of the protein, and then we beg to differ and disagree with Rob Tika, who says that this is the extension of the fibril body. We think it's not. It's really a flexible tail. So I think with this slide, I would like to more or less finish the presentation. Again, I would like to acknowledge the contribution from all of my co-authors. Last Spinous conference, we were able to come here, all of us, and each and everyone gave a talk. Again, we acknowledge the seminal contribution from our university, then the support from Russian Science Foundation. Again, Vladimir Ivanovich served as an inspiration for all of this. The resource center, so without the spectrometers, we couldn't do our work. And that's the collection of the pictures. So I mean, we run the electrophoresis. We do the we have those GPU-based workstations, very, very powerful and high-end Teslas. That's very good. And in terms of, travels, last summer we had the chance to travel all together, the entire lab, to Jyväskylä, to Finland, and we were at the Scandinavian NMR Society meeting. And here is Sebastian and Sergey explaining their work to Art Palmer from Columbia and Dorothy Kern from Brandeis. And so with these things, I would like to finish. I'd like to mention also at the end that we are looking for graduate students and we're looking for postdocs. So if that's of any interest, I'd be very happy to talk to you. I'm myself very interested. I've been very interested in being here for the poster session and seeing the, all the posters and talking to many of you. I went through the book of abstracts, but I won't be able to do here because my daughter turns 4-year-old, so that's her birthday that I can't miss. But I'd love to, to talk to many people in the audience and discuss your research. And so with this, I'd like to finish and thank you for your attention.
Thank you very much for this interesting, nice presentation. We have still time for questions and comments. Please, may I ask you? Please, Colin.
Thank you for your presentation. And I have a question. I don't know, I don't believe that is an important question, but I don't understand if it is important or not. The problem is related to the gradient along the sample. You have a gradient along the sample. Do you invert the sign of the gradient to prove that it's not independence of the gravitation influence in the mass of the fibril and monomer and so in the diffusion?
Mm-hmm. So the way that we apply the, I know what you mean. So the way that the gradients are designed, we basically have a gradient, and then there is a 180-degree pulse, and then there is an inverse gradient.
Yes.
But so those two gradients combine, that's how the way that Kurt Woodruff has introduced it. Now there is always this issue about the convection, right? So it might be diffusion, it might be, it might be also that the bottom of our NMR tube is heated a little bit more and there is this convection flow, and so then you're not measuring diffusion, you're measuring basically convection. So one thing that we always are careful to do, we use those Shigemi tubes with little volumes, little sample volumes. Yeah. And there is a nice review paper by William Price and Concepts in NMR, kind of saying, okay, use Shigemis, then you're going to be safe from convection. Then the other rationale why we think that we have no problems with convection is that what we measure as a diffusion coefficient for monomer is exactly the textbook value. So there is a graph that's kind of empirical graph with several dozen proteins plotted there and their diffusion coefficients versus mass. And what we find is really corresponds to this monomeric sub-35 nm falls right on the line. And likewise for the fibrils, we do see that for the fibrils, there is no diffusion at all. So if you imagine a convection, then fibrils would probably go with the flow, even though they are big, right? But convection would carry them also. So I am hoping that, I'm thinking that probably there is no reason to think that convection is a big issue.
Okay, thank you.
Okay, thank you. Further questions, comments, please? I have a question. In the beginning, you have shown amino acid sequences which are identical, although the behavior of these parts is different, probably due to the diaeder angles and the configurational change, if I understood you in the beginning very well. But now you have shown the motion of the fibrils. Could it be that this fibril motion is also influencing the diaeder angle? It means the configuration of this complex sequences of amino acid, and to change the properties, the overall properties of these parts.
So I think in the first part, the point that I was trying to make is with amino acids is that there are those Ds and Ls, and they are natural and unnatural, right? But looking at the dynamics of the fibrils, so what happens with it, I believe, would be, if we have a schematic here somewhere, close, this one. So if you look at the dynamics, you can use solid-state NMR to look at the dynamics of those. And that's not different from folded protein. So there is a little bit of you know, harmonic kind of axial fluctuations there and things like that. But the amplitude is limited. That would be 10 degrees, in the ballpark of 10 degrees. And the side chains move, but that's what you see in, you know, ubiquitin or BPTI kind of textbook proteins. And the tail is apparently the you know, the disordered tail. So that's floppy, and that is a disordered protein. That's like a worm that's just wiggling and then twisting and so forth. In a sense, we tend to believe that, of course, this fibril forming body is more important, and even small details of dynamics can be consequential, because the way that the fibrils are formed in our brains, no one knows what triggers the process, and the process is extremely, extremely slow. So there got to be kind of a boundary that, you know, below this boundary you stay healthy, but a little bit of this butterfly wing effect, the fibrillation, the fibril aggregation starts, and you get this deposition in your brains. So there, you know, the mutations definitely matter, because there is such thing as familial Alzheimer's disease, and the details of dynamics may matter, and oxidation of methionine is known to be important for Alzheimer and things like that, and, you know, copper coordination and so forth. So some minute details here can be relevant for eventually, you know, designing the cure for Alzheimer's disease.
Thank you very much.
Thank you.
If there are no further questions, I'd like to thank you once more for this excellent presentation. And now we are looking to the contributions of your co-worker, and we are interested to also see